Hi, my name is Maddie Hood. I am in Dr. Karen Kettenring's lab, which is the Wetland Ecology and Restoration Lab. And my research is using plant traits in successional revegetation to restore Great Salt Lake wetland plant communities. So in the past 200 years, we have lost over 50% of our wetlands in the United States. In the Intermountain West, large wetlands are even rarer, but they provide critical habitat for wildlife, including millions of migratory birds that use Great Salt Lake every year, as well as billions of dollars in ecosystem services annually at Great Salt Lake alone. And these remaining wetlands face increasing challenges due to irrigation and drought withdrawals, as well as invasive species spread. One of these invasive species is Phragmites australis. Phragmites is a large-scale invader in the United States from Eurasia, and it's problematic because it turns these complex wetland plant communities into dense monocultures of grass that wildlife have a really hard time getting through. And in our area, it's really common to treat for Phragmites with herbicide and biomass removal, but revegetation after these efforts is pursued far less often. And when it is pursued, these revegetation efforts are met with variable success due to these same challenges. So some of these motivations for restoration include building drought resiliency and invasion resistance into our plant communities, as well as general habitat recovery of our wetlands. So the overarching question that guides my research is, what are the best practices to restore these native wetland plant communities in the Intermountain West? And within that, I have some specific questions. So how does seeding compare to planting when revegetating wetlands? And when I'm talking about planting, I'm referring to these plugs and sod mats. And specifically in my research, I'll be using plugs. But essentially, we partner with North Fork Native Plant Nursery, which is up in Idaho. And we'll provide them with native seed, which we gather in the field up to a year, sometimes more in advance. And they grow them out into these plugs over the course of the year. And germination is one of the most limiting stages in a plant's life. So by growing them out in the greenhouse, we can essentially bypass that stage and have greater success in the field. And can seeding be combined with planting for greater plant establishment in a successional vegetation framework? And this successional vegetation framework, typically in a terrestrial system, this will refer to planting annuals in the spring, so in May or so, followed up with a perennial planting later in the summer. So these annuals are serving as sort of a crop cover. They can preempt resources and potentially push out invaders by gathering those nutrients first. And by establishing earlier, they have a better chance of surviving harsh environmental conditions that they may face later in the summer. So some more questions I have are, what species are predicted to perform well based on their functional traits? And can functional groups establish greater native veg when applied to this successional planting framework? So what are functional traits and functional groups? Well, essentially, they're just a larger way to group plants than just genus species. So there are many different plant traits that we can gather and that we use currently, which include things like root density. Is it a really dense root mat, or is it just like a single tap root? Life form, forb or graminoid. Seed mass, is it a dense seed that will sink to the bottom, or does it float on water surfaces? As well as your height in the canopy. And when you combine these into functional groups, you can choose for specific plant functions. So for example, if we go back to that root density, if you choose a group that has a higher root density, then this can again preempt those resources by taking up more space on the landscape, as well as it has drought implications because they can reach deeper into the soil and get more water. So to address these research questions, I have both a greenhouse experiment and a field experiment. The greenhouse experiment will identify species that perform well in drought and with high growth potential, specifically against Phragmites, and collect trait data to inform functional group seed mixes. I'll then be testing these seed mixes in the field and also determine the potential benefits of successional planting. Again, here are my objectives for the greenhouse experiment, and I just want to highlight that it is important to choose for these species that will perform well in drought because of the increasing drought we've been seeing in this area and the prolonged drought, as well as the decline of the Great Salt Lake, which has resulted in a 50% reduction in lake volume, which creates these drought conditions where they might not otherwise have been. So in my greenhouse, I tested 38 different species. These were a mix of graminoids as well as forbs. And the forbs were chosen by the lab and were noted for their success in the wild and for their potential for use in revegetation. So I tested these 38 different species at 2 different water levels. The first was a saturated water level, which is where the water surface is the same level as the soil surface. And the second is a drought where the water is 10 centimeters below the soil surface. There were 3 replicates of that, and the plant responses I measured were in biomass, height, as well as growth rate. And this is not a Lowe's ad, but these are my reservoirs. So I have a 1-gallon nursery pot with which I seeded the individual species in each pot. And then that Lowe's bucket was serving as the reservoir. Wetlands are ground-fed systems, so this was imitating that. For data collection, first I tracked emergence of every seedling that came up. So any seed that germinated and put up a shoot, we marked it with a stick in the date of emergence. And then after a set period, I assigned 6 seedlings within each pot to either a 14-day harvest, which is from the date of emergence, or a 28-day harvest, which is my final harvest. And that was to establish a growth rate. I also collected other above and below ground plant traits, which include biomass, height, as well as root elongation rate and root density, which is what you're looking at here. This is actually a scan from my experiment. So I separate the shoots from the roots, we scan it, and then we have software that will individually measure each one of these and combine it into a total root length, which I can use. And this just finished up on Monday, so I'm definitely in the preliminary phase, but so far just visually, there's been some pretty clear differences between species and these root densities. To analyze this, I plan on using mixed effects models on each of my responses, which are the biomass, height, and growth rate, with the fixed effects being the species times the water level. I also plan to do some structural equation modeling, looking at the direct and indirect effects of plant traits on each of those responses. So for my field experiment, I will be looking at which functional group planting combination has the greatest survival and species richness, and if successional planting can increase native canopy cover compared to just a single planting effort. To do this, I established 2 different seed mixes. The first is a forb mix. It is full of early successional species. These were chosen for their larger root systems, which I found in my greenhouse study. And these species, again, we're thinking annual, so they establish quickly. And this, again, has implications for drought and invasion resistance. You can preempt those resources, and you can be a larger plant for when you experience those extreme drought conditions that we have been experiencing. And seed mix 2 is a graminoid mix. This is late successional species. These plants typically take longer to develop, but once they do develop, they persist year after year on our landscape. And plug mix 2 will be the same species as the seed mix 2, so the same graminoid species, but we've bypassed that germination stage. So I will have 2 different planting efforts. The first will be in May, and the second will be in August. And first, my first treatment will just be nothing on the landscape, and this is just to monitor the background noise and just see what comes up in our seed bank. Second, I will have nothing in the spring, followed with just the plug mix in the fall. The next few treatments, I will do either that forb mix or that graminoid mix with no follow-up planting. Then I will have the forb mix followed with either the seed mix of the graminoid species or the plug mix of the graminoid species. And lastly, I will combine those 2 seed mixes with no follow-up planting And I just want to note that's typically what has been done in these revegetation efforts, where you just put all your seeds together and just put them out on the landscape all at once. So I plan to monitor percent canopy cover, survival, and species composition, and do some more mixed effects modeling on each of those responses, where the fixed effect will be the seed mix times that planting combination, which we just looked at. So where is this work being done? It's going to be at Farmington Bay Waterfowl Management Area, which, as you can see, is kind of on that southeast side of Great Salt Lake. This is the outline of the area, and the X is the Teal Lake unit, which is actually where I will be revegetating. And Teal Lake is an actual lake that will fill up with spring runoff, and then it'll be slowly drawn down throughout the summer. So conditions will be flooded in the spring, followed by really dry conditions later in the summer. They've done Phragmites treatment in this area since about 2018, using both aerial spray as well as backpack sprayers. And they come back in after to remove the biomass, which prepares it for seeding. Despite this treatment, there's still coverage of 20 to 60% Phragmites, depending on where you are in the Teal Lake unit. But this is pretty typical conditions that restoration practitioners actually face in the field. So this work is being performed on Great Salt Lake. It has implications statewide, and this work will be shared with managers statewide, as well as throughout the Intermountain West. It will create a better understanding of these best seeding and planting practices in this functional trait framework, as well as successional planting. And then it will increase the drought resiliency and invasion resistance of our wetland plant communities. Thank you. Yeah.
Sorry if I missed this, but other than drawdown, are you trying to capture any environmental variability in your treatment? Local differences in moisture or?
I don't think that will be in the scope of my experiment, because it is so difficult to capture that with everything going on with the treatments, unfortunately. Yes.
What's the source water in your reservoirs?
Could you repeat that?
What's the source water for your reservoirs? Tap water, or
It was tap water, but I did add fertilizer to the soil mix. And that was filled up every day. So it was getting like continuous, like colder water inputs, I'd say. Okay, thank you.